so it's a competition like no other. The Connecticut Science and Engineering Fair features impressive projects by some of the top middle and high school students from around our state. And today we're lucky enough to have 3 of the finalists. Mallory Matfis is from Greenwich High School, Delaney Kehoe is from Glastonbury High School, and Jordan Henk is from Amity Regional High School. Ladies, thanks so much for being here. This is pretty exciting. These projects have come quite a long way since I was doing a science fair. We won't say how long ago that was, but I understand this is the 65th year, and there are about 12,000 students that start off. It's narrowed down to 600 and then 150, and then you guys are the top tier here. Mallory, why don't you tell us a little bit about what people can expect at the fair?
So you can expect anything from engineering to biology to plant life, dressings, medicine, really the full wide range of science.
Now, where are people coming up with these ideas? Is it kind of a free-for-all, whatever you have an interest in?
Yeah, really what people's interests are in.
Okay. Delaney, I want to ask you, I know that you guys are also finalists for the Connecticut Technology Council's Women in Innovation Awards. That's kind of a cool honor.
Yeah.
I know that there's something coming up later this week.
Yeah, next Tuesday night we have an awards dinner at the AquaTurf where we'll figure out who wins out of our youth innovation category.
Are you guys in competition right now, kind of?
Yeah.
Oh, all right. Well, congratulations to all of you. Let's talk about the projects. Mallory, we'll start off with you.
Yeah.
What are we looking at here? Break it down for us.
So this is micro-remediation of PCB soil contaminants with Paratus austratus. And this is kind of just, I used a fungus to help get rid of PCBs in soil.
Okay.
And it was successful.
And this was last year's project.
Yes.
This year's project is at home drying still.
Yes.
What is the topic this year?
This year it's promotion of wound healing via a novel hydrophilic dressing. So basically what that is I made a dual component wound dressing, kind of like a Band-Aid,
okay,
where there's 2 different components in it, one that will help grow skin back and one that will stop infections from starting.
And what were the results?
Very, very successful.
Really? Now, what made you choose a topic like that?
My mom's a dermatologist, so I've been growing up around wounds my whole life.
So last year you earned 4th place, so you're
Yes.
trying to get a little higher this year?
I earned 4th place at Intel ISEF, which is an international fair.
Oh.
And hopefully I'll make it back there again.
All right, sounds good. And do you have any advice for anyone that might be involved in this?
Just keep working hard. No matter, you can really do anything if you put your mind to it.
Very good. Well, thank you. Delaney, we'll talk about your project now. Give us some of the details.
So my project is, the goal is to monitor the pollution in the northwestern Atlantic ecosystem through immune response systems in the livers of sharks. So we look at these cells called melanomacrophages, and they appear in the shark's liver when the shark encounters a toxin.
Okay.
So we look at the occurrence of the melanomacrophages in the livers with a microscope, and we record them. And then the results that we have are added to an archive of information from the past 15 years, and it will eventually just track the amount of toxins that are in the ecosystem.
And what are you finding so far? What's the result been?
So far, it's slowly increasing.
Okay. Do you plan on doing marine biology when you get to college?
I'm not quite sure what I'm going to do yet, but mostly just, I like biology, so something in there.
Something along those lines. All right, very good. Well, good luck to you. And Jordan, let's talk about yours on the end here. Break this down for us.
Okay, so my project is called The Prediction of Cancer Tissue Origin based on molecular sequencing evidence using a machine learning-based classifier algorithm.
Okay.
So the way pathologists analyze cancer tissues is they end up opening up the patient, taking out the tissue sample, and looking at this under a microscope. This is a very visual way of doing so. But another way to be doing so would be instead, when you have that tissue sample, would be to sequence its DNA, and then you can use this DNA to tell you a lot of information about the types of cancers. So I did this using a machine learning algorithm because there's a lot of chaos and disorder within the DNA, and so you have to kind of find some order to it.
Sure.
And so I used a decision tree algorithm for this, and it's kind of like the game 20 questions, where it starts out with you're trying to find an answer, and so it asks a series of questions until it gets the answer. So in my case, it asks questions on whether or not the gene is mutated. So if the gene is mutated, it will go down a certain path, and it will ask more questions about different genes being mutated.
Okay.
And if it's not mutated, then it will keep on asking about different genes. And so at the end, I ended up looking at it for sensitivity and specificity. So Those are two things that are very important. Sensitivity is the ability to predict correctly that you contain the disease, and specificity is the ability to predict correctly that you do not contain the disease. Oh. And so I found that there was very high specificity but varying sensitivity within the data. Okay. And in the end, I found that there's a 67% accuracy rating when you looked at metastatic cancers. So this is cancers that have, 67% when you looked at non-metastatic cancers, which are cancers that have not moved within your body. Okay. And they've stayed in the same location. And so this is 60% higher than the 7% accuracy rating you get if you were to randomly predict just based on the population. Got it. While looking at metastatic cancers, so these are cancers that have moved from one location in your body to another location, there's a 44% accuracy rating, and so this is still higher than the random predictiveness. And so what this really shows is that genes can be genes can tell you what kind of cancer you have and can give you information so that you can come up with better diagnosis for your cancer.
Wow, you guys are all going to be scientists. I'm so impressed by these projects. Fantastic. All right, let's put some information up on the screen. We should mention that News 8 will be participating in this year's science fair. Our own meteorologist Quincy Vagel is going to be one of the judges. Gil Simmons will be doing the noon broadcast there live on the fair Thursday, March 14th. We're going to have the weather lab out there. Fair participants will be writing about the work on wxedge.com, so you can check that out. Again, it's the 65th annual Connecticut Science Engineering Fair today through March 16th at Quinnipiac University. Go to ctsciencefair.org for more information. Ladies, you blew me away here. I'm so impressed. Good luck to all of you.
Thank you.
All right, stay with us. More Connecticut Style after the break. We'll be right back.